This is the third of a set of presentations illustrating the Stop Thalassemia project of the Cure to Children Foundation. Bone marrow transplantation for severe thalassemia prospects. In this presentation, we'll summarize the global access to compatibility testing, how this is possible, using advances in supportive care to downstage high-risk patients and decrease transplant-associated risk so that more and more thalassemia patients can actually safely access this definitive cure for thalassemia, increased donor availability. In particular, we discuss the mother as a bone marrow donor, briefly discuss gene therapy versus bone marrow transplantation, and illustrate the Stop Thalassemia vision of this project. Thanks to a simple modification of the HLA typing, which is a test to assess compatibility, for example, among siblings of patients with thalassemia, it is now possible to do HLA typing globally by a very simple buccal swap from which DNA is obtained, and HLA typing is done in a central, cost-effective, and very reliable way. So this has made a huge difference in terms of extending the potential to cure children with thalassemia. The other important step forward is the advances in supportive care, particularly chelation therapy, but also a simple and inexpensive drug like hydroxyurea, which can revert many thalassemia patients to a low-risk situation. In other words, can remove a lot of the iron overload, as well as decrease liver or spleen enlargement. This is so-called downstaging prior to bone marrow transplantation. So that we know that with increasing iron, there is increasing organ damage. But nowadays, with improved therapy to remove iron from the body, we can actually conceive that most patients with thalassemia could be reverted to a low-risk situation by removing a lot of the iron overload. Then the issue of extending bone marrow transplant access in terms of increasing donors. So as discussed in the first part, the majority of children may not have a matched sibling, and it is becoming increasingly evident that probably the most realistic alternative is to use a partially matched family donor, particularly for children, for example, the mother, who's normally 50% compatible. In the experience in a closely related disease, which is sickle cell disease, actually the use of haploidentical bone marrow donors, so partially matched family donors, had resulted in a very low mortality rate, very low graft versus host disease, and unfortunately the problem is still graft failure. Almost 50% of patients may reject their graft. But there are ways that this can be improved, and so most likely in the future, many more patients with thalassemia will be able to have a transplant even if they don't have a matched related donor. Gene therapy, many people probably don't realize that there is not a major difference between transplantation and gene therapy. Actually, gene therapy is a form of gene-modified autologous stem cell transplantation. So that nowadays that gene therapy is starting to be offered on an experimental basis. The process still implies the administration of a preparative regimen, which is actually not very different from what is used in bone marrow transplantation. So, gene therapy is a form of autologous bone marrow transplantation that employs gene-modified autologous stem cells, while the classical bone marrow transplantation is an allogeneic transplantation that uses hematopoietic stem cells as the vector of the normal gene. The Stop Thalassemia vision, so survey, treatment, outreach, and prevention of thalassemia. So the Q2 Schindler Foundation, based on its experience, and would like to develop multiple independent bone marrow transplant centers in the hemoglobinopathy belt, sharing protocols and procedures and non-profit objectives. So the idea is to have a global network of centers offering the cure of thalassemia by bone marrow transplantation and using the software to also promote counseling, screening, and prevention. So the role of third-party international protest organization is that to foster use of common protocol and procedure, maximizing quality assurance, promote screening and prevention, reassure patients and professionals during startup. We have seen how, even in the startup phase, within close collaboration, very good results can be obtained from the very beginning. Decrease unnecessary expenses. This is also very important because a high cost of transplant means less accessibility and more children that will not be able to become adults because they don't have access to a cure. Increasing scientific visibility is very important for the credibility and the prestige of partner centers. Facilitate quality assurance, support fund development, sustainability strategies, and this is the objective of the Stop Thalassemia project, provide network referrals. So in conclusion, thanks to the advances in both supportive care and bone marrow transplant, most individuals with thalassemia have the potential for a safe and accessible definitive cure. Thank you for watching this video, and I hope you will support the Stop Thalassemia campaign from the Q2 Schindler Foundation.